Welcome to Legumes in Space.
Hello and welcome. Today I'm joined by Michael Wilkinson, who is the ExoLab 8 principal investigator, and he's also a math and science teacher in New York.
Hi, it's great to be here, Lori. Lori's our Director of Communications at Magnitude, and we've got some great news for everybody this week, I think.
So Michael, why don't you go through some of the highlights from the past week?
Okay, it was kind of exhausting. We started out on Saturday afternoon launching to the space station on the Katherine Johnson. Soichi Noguchi captured the Cygnus really early in the morning Eastern time over the Atlantic Ocean with the Canadarm. And then Houston Robo team commanded the capsule in for a berthing. And then on Tuesday midday, astronaut Kate Rubins, offloaded the ExoLab from the Katherine Johnson and installed us in the U.S. National Laboratory in the Destiny module.
And we got some initial images when she powered it up. And I bet we've seen some interesting things in those, aren't we?
Yeah, there's some really interesting stuff. And I want our students to really look closely at those images, blow them up and look all through the test tube, see what you notice, see what you wonder. These are the really important beginning stages of the experiment and could shape the way the whole experiment plays out. So these initial observations are super important, and even more important are all the questions you might have when you look at this. I know I've got about 1,000 questions having just looked at one image, so I can't wait to hear the questions from everybody else, because that will shape what the important tests are that we do going forward.
Because this is a demonstration project. This is the first time we're trying growing clover in space to achieve nodulation, but this is also the first time that this particular cube lab has flown. So we're looking at both the design and engineering, as well as on the science side as to what's happening. So
whose crazy idea was that, Lori? oh yeah, that was my idea.
Well, you know, we have to dare mighty things to try new technologies in order to explore these science topics. Especially, we know our clover likes to germinate rapidly, and so we're seeing some of that in these very early images. And so I think, Michael, this might be a good time for you to talk about lesson 4 with all the data and observations and what students and teachers can expect to see and what they really need to pay attention to and notice, what measurements quantitatively, qualities to take note of, and be sure to get all those details recorded in the right way. So why don't you tell us some about lesson 4 on the platform?
Sounds like a good idea.
Once your experiment's all set up, it's time to start making observations and recording them. Lesson 4 is going to help walk you through that whole process. We'll explore what it actually means to make observations, why we make observations, as well as give you a form each week where you can report those observations. Each week it's a new form, and you'll choose a representative plant from your experiment to observe and document. This form will walk you through that process, and there are some guiding questions at the top. To actually see your experiment, remember, you'll go to the experiment dashboard. And once you get there, there are 2 images. The one on the left is your experiment, and the one on the right is the experiment on the International Space Station. There's some information about the environment of each of the labs that you'll want to take note of. But the main thing that we're doing right now is, how are these seedlings growing and developing? So you'll choose one of those seedlings in your experiment, and using the digital notebook, you'll fill in that table with each of the pieces of information that we're asking for. Make sure that you indicate which specimen you're recording your observations for. There are 2 tubes, left tube and right tube, and seedlings 1, 2, and 3 in each of the tubes. I'm really looking forward to seeing what you see, what you notice, and what you wonder.
So Lori, those are the main points on the experiment that we need to pay attention to. Can you tell all of our participants how to get their ExoLab set up and running so that they can share their data with us?
Absolutely. So it's time to start firing up those ExoLabs. So let's go through some of the instructions. Once you have registered your ExoLab, connected it to power and to Wi-Fi, you will go to your Magnitude Classroom dashboard menu, select ExoLab, click on Start Experiment. In the ISS Lab drop-down menu, scroll to the bottom and select ExoLab 8 ISS, then give the experiment a name. The start date is today, Friday, February 26th. So you select whatever day that you're beginning on. I want to keep this experiment public, so I will not check the box. We are running a dual tube experiment. And then the configuration options. On the left tube setting, we will choose white light. This is a change from our original protocol. We'll keep the light intensity at 10 and the photo period at 16. For the right tube, we'll select magenta light, light intensity of 10, photo period of 16. And then we click Save Experiment. Once the ExoLab begins taking photos and logging data, you will see the information on the experiment page. If you want to compare with other ExoLabs around the world, once they've launched today, you can begin to compare those by looking through the data and seeing all the ExoLabs that have been started up for this experiment. So one other thought as we're wrapping up Black History Month this February is the significance of arriving to the International Space Station on the SS Katherine Johnson. And as you may or may not know, Katherine Johnson was instrumental in doing the math calculations to get John Glenn into orbit in the 1960s. And so we see significant contributions from African-American men and women throughout history in regard to space flight. And even our legumes have some history there. Michael, tell us about another historical figure.
Yeah, I've been fascinated with legumes since I was a boy, growing up in Iowa, farm country. And a particular legume expert is also from there and did some of his formal study there, and that's George Washington Carver. He was always a real inspiration to me in terms of how he went about just exploring this class of plants, legumes, and how their function in the ecosystem. But he also was an artist. He was a very accomplished botanical illustrator, and he ended up studying at Iowa State University, where I ended up going for my degrees. And so it was always, just so important to me that any work I did really good illustration documentation of it. And now the photography work that I do in close-up photography and the microscopy all is really an offshoot of that work that he kind of put out there and caught my attention as a young boy. So it's really exciting to have those kinds of connections in this work.
Definitely, and we see some of your artwork in some of these lessons. So for the students, it's really important to take what they're observing and to use those creative skills too, to draw out and diagram and notice those pigments and colors. And so we can learn a lot from those illustrations, and that's great inspiration from George Washington Carver and those beginnings with art, and then on to studying rejuvenating the soil. And that's a key part of what we're investigating too. So we have great connections to these phenomenal historical figures who've contributed to science and to space exploration.
Standing on the shoulders of giants.
That's right. Well, that does it for this week. Our highlight of what happened with ExoLab 8 on the International Space Station. We look forward to having you join us live for our session on Thursday, March 4th at 1 p.m. Eastern. If you would like to submit any questions to be asked during that session, you can send them in advance to hello@Magnitude.io. And if you'd also like the Zoom link, you can send an email to that address as well. Until then,
Stay
signing
curious,
off.
everybody.
Stay curious.